For most of the 20th century, scientists believed they had the story of human evolution figured out. A clean, straight line running from ancient ancestors to modern humans, one species rising, every other branch of the human family dying out along the way. Simple, solved. Then, in 2010, that entire picture cracked open. A team sequencing genetic material recovered from ancient bone discovered something that shouldn't have been possible. Modern humans hadn't just crossed paths with other human species on our way to becoming the only ones left. We had interbred with them, and their DNA is still sitting inside us today, in your genome, right now, whether you've ever thought about it or not. Fast forward to 2024. Geneticists in Japan sequencing thousands of modern genomes in one of the most detailed studies of an Asian population ever conducted, uncovered something that didn't just confirm the old story, it complicated it. Buried inside the DNA of people living in Japan today are fragments of genetic code from populations that vanished tens of thousands of years ago. Populations that, in some cases, left behind almost no bones, no tools, and until very recently, no real physical trace of ever having existed at all. This isn't ancient history sealed off behind a museum glass case. This is present tense biology. This is sitting inside the cells of tens of millions of people walking around right now. And once you understand what it actually means, you start to look at the whole idea of what a modern human even is a little differently. If discoveries like this are the kind of thing you want more of, stick around and subscribe. Because this particular thread runs a lot deeper than most people realize, and by the end of this, the story of who actually counts as fully human is going to look considerably or less settled than it did 5 minutes ago. Let's rewind to where this entire story actually starts, because you can't understand what happened in Japan without understanding the discovery that made anyone think to look for it in the first place. For most of the last century, the reigning explanation for human origins was called the out of Africa model, or more specifically, the replacement version of it. Homo sapiens evolved in Africa roughly 300,000 years ago, then spread outward across the planet, and everywhere we went, we simply outcompeted and replaced whatever other human species we found there. Neanderthals in Europe? Gone. Various archaic populations across Asia? Gone. We were the last ones standing, the sole survivors in a story that had, up to that point, only losers and one winner. That was the textbook version, and for a long time it held up reasonably well. But by the 1990s, geneticists mapping human DNA from populations around the world kept noticing small inconsistencies, stray genetic variations, that seemed older and more distinct than they should have been if every living human's ancestry really did trace back to one single, unbroken African lineage with no outside contribution at all. Then, in 2010, a team led by geneticist Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology did something that had never been done before. They sequenced a complete Neanderthal genome, not scattered fragments, the entire thing, pieced together from bone recovered from a Croatian cave. When they compared that genome against the DNA of living humans, they didn't find vague, distant similarity. They found exact matches, specific blocks of Neanderthal genetic code sitting directly inside the genomes of people alive today. Europeans and Asians carried somewhere between roughly 1% and 4% Neanderthal DNA. Sub-Saharan Africans carried almost none, a pattern that made perfect sense once researchers worked out the timeline. Sometime between roughly 50 and 60,000 years ago, as early modern humans expanded out of Africa into the Middle East and beyond, they encountered Neanderthal populations already living there. And they didn't just meet them, but they had children with them, hybrid children who survived, who were raised, who had children of their own, generation after generation, until fragments of that ancient DNA were still riding along inside human genomes tens of thousands of years later. Pääbo would go on to win the Nobel Prize in Physiology or Medicine in 2022 for this body of work, a recognition of just how completely it rewrote the field. The old textbook picture, one clean line, one surviving species, was wrong. We hadn't simply replaced our closest relatives, we had absorbed them. Neanderthals, though, turned out to be only the first thread in a much bigger tangle. In 2008, Russian archaeologists working in the Denisova cave in the Altai Mountains of Siberia recovered a tiny fragment of a finger bone, so small it could have easily been overlooked entirely. It belonged to a young girl who had died roughly 40,000 years ago. When Pääbo's team sequenced her DNA expecting to find another Neanderthal, they found something else entirely, a previously unidentified human lineage. Researchers named this new population Denisovans, after the cave where that first fragment turned up. It's worth pausing on just how remarkable that identification actually was at the time. A single finger bone, barely large enough to see clearly without magnification, turned out to contain enough preserved genetic material to reveal an entirely new branch of the human family tree. one nobody had previously known to look for, entirely because the technology to read ancient DNA at that level of precision had only just become available. Before that moment, the working assumption in the field was that the archaic human landscape of Eurasia consisted of Neanderthals in the west and pockets of Homo erectus descendants scattered further east. The Denisova fingerbone blew a hole straight through that assumption in a single afternoon of lab work. And here's what made them genuinely strange to work with scientifically. Despite clearly being a distinct, widespread population, Denisovans left behind almost nothing in the way of recognizable fossils, no complete skeletons for decades. Just that fingerbone, a handful of teeth, and eventually a jawbone recovered from a cave on the Tibetan plateau. For close to 15 years, nearly everything scientists knew about Denisovans came exclusively from their DNA, extracted from a small handful of bone fragments, because nobody could say with confidence what one of these people actually looked like. And that DNA told an expansive story. Denisovans weren't some tiny, isolated group tucked away in one corner of Siberia. Genetic traces of them stretch across a huge swath of Asia and out into the Pacific. Indigenous peoples of Papua New Guinea and parts of island Southeast Asia carry the highest levels of Denisovan ancestry recorded, anywhere on Earth, in some populations reaching close to 5% of their entire genome. Somewhere in the deep past, modern humans met Denisovans too, and exactly as with the Neanderthals, they had children together. That brings us to Japan, and to a study that landed in 2024 and genuinely reshaped how geneticists think about one of the most heavily studied populations in Asia. For years, the accepted picture of Japanese ancestry was fairly simple. A dual origin model built around two main groups, the Jōmon, an indigenous population of hunter-gatherer fishers who lived across the Japanese archipelago for thousands of years, and a later wave of rice farming migrants who arrived from the East Asian mainland and became closely associated with the Yayoi period. Two source populations blending together over time to produce the modern Japanese gene pool. That was the model everyone worked from, right up until a team of researchers at Riken's Center for Integrative Medical Sciences, led by geneticist Chikashi Terao, alongside lead author Xiaoxi Liu, decided to test it against the most detailed genetic dataset ever assembled for the Japanese population. Drawing on whole genome sequencing of more than 3,200 individuals collected through Biobank Japan, spanning 7 regions from Hokkaido in the north down to Okinawa in the south, and publishing their findings in the journal Science Advances in 2024, the team found that the old 2 population story didn't fully hold up. Hidden inside the data was a signal of a third ancestral component, a population with genetic ties to northeastern Asia, one the researchers connected to the historically documented Emishi people, adding real weight to a tripartite origins theory that had first been proposed a few years earlier in 2021. As Terao put it, the Japanese population isn't nearly as genetically uniform, as most people assume. Fine-scale analysis revealed structure that mapped surprisingly cleanly onto geography, different regions carrying subtly different signatures, depending on how these 3 ancestral streams mixed together across the archipelago over thousands of years. But the tripartite ancestry finding, remarkable as it was on its own, wasn't the part of the study that really stopped people in their tracks. While mapping the genome in that level of detail, Terao's team also went hunting specifically for fragments of DNA inherited from Neanderthals and Denisovans, and they found 44 distinct archaic DNA regions still present and active inside modern Japanese genomes. 42 traced back to Neanderthal ancestry, and 2 traced back to Denisovan ancestry, the overwhelming majority of them unique to East Asian populations, meaning they don't show up in this particular combination anywhere else on the planet. Terao has said the team is specifically interested in why these ancient genetic fragments got integrated into the modern human genome in the first place, and, more importantly, why evolution bothered to keep them there for tens of thousands of years instead of quietly erasing them the way it does with most neutral or harmful mutations over that kind of time scale. The answer, in case after case, seems to be that these fragments weren't neutral at all. They were doing something useful. Start with the Denisovan linked findings, because one of them connects directly to a health condition affecting millions of people today. One of the 2 Denisovan-derived segments identified in the Japanese genome sits inside a gene called NK6-1, a gene already known to researchers studying type 2 diabetes. The archaic version of this gene appears to influence how the body regulates blood sugar, and researchers have suggested it may even affect how some individuals respond to semaglutide, the drug widely known under brand names used to treat diabetes and, more recently, obesity. Think about that for a second. A genetic variant passed down from a Denisovan ancestor tens of thousands of years ago may be quietly shaping how a diabetes medication approved in the 21st century actually performs in a specific patient's body. That is not ancient history sitting inert in a genome somewhere. That is active, functioning biology with direct, present-day medical relevance. The Neanderthal-derived segments told an even broader story. Researchers connected different regions of that Neanderthal DNA to a whole cluster of modern conditions, including coronary artery disease, rheumatoid arthritis, prostate cancer, and Graves' disease, an autoimmune disorder affecting the thyroid. This lines up with a pattern researchers have already documented elsewhere in the world, a cluster of Neanderthal-inherited genes on chromosome 3, present in roughly half of all South Asians, has separately been linked to a significantly higher risk of severe respiratory failure during COVID-19 infection. A genetic legacy from an extinct relative directly shaping outcomes during a 21st century pandemic. None of these inherited archaic genes are simply harmless genetic fossils sitting quietly in the background. They're active, they're doing something measurable, and in some cases what they're doing has real consequences for modern medicine, meaning treatments that work well in one population might need to be understood differently in another, purely because of which ancient human lineages happen to contribute to their ancestry. There's a companion piece to this story worth understanding too, because it explains a puzzle that used to genuinely frustrate researchers, namely, why certain populations carry so much more Denisovan ancestry than others despite living relatively close together, geographically. In 2018, geneticist Sharon Browning and her colleagues, studying the genomes of people from Papua New Guinea, found something unexpected. Evidence that Papuan Denisovan ancestry didn't trace back to just one archaic population, but to two genetically distinct Denisovan-related groups. One more closely related to the Denisovans, known from the Altai region of Siberia, and another so genetically divergent from that Siberian lineage that it looked almost like a separate population entirely, one that had likely been isolated from the Siberian group for hundreds of thousands of years before either of them ever encountered modern humans. That finding matters enormously for understanding Japan's own archaic ancestry, because it demonstrates something researchers now take seriously across the whole field, that Denisovans weren't a single uniform population quietly waiting in one cave. In Siberia, they were a widespread, genetically diverse group of populations scattered across huge stretches of Asia, likely including branches that modern researchers haven't fully mapped yet, each contributing their own distinct genetic signature to the modern populations they eventually interbred with. Which brings us to the single strangest twist in this entire field, the case of a skull that sat completely misunderstood in a museum drawer for the better part of a century. In 1933, a Chinese laborer working in Harbin, in the country's northeast, uncovered an enormous, strikingly heavy human skull, and rather than hand it over during the Japanese occupation of the region, he reportedly hid it down a well, where it stayed hidden until his family finally recovered and donated it to researchers in 2018. When paleontologists formally examined it in 2021, they were struck by just how unusual it was. A massive brain case roughly comparable in size to a modern human's, paired with a broad, heavy face, thick brow ridges, and other features that didn't cleanly match Homo erectus, didn't cleanly match Neanderthals, and didn't cleanly match us either. The team studying it, led by paleontologist Chiang Ji, proposed it belonged to an entirely new species, which they named Homo longi, nicknamed Dragon Man, after the region where it was found. For years afterward, the identity of Dragon Man remained genuinely unresolved. DNA extraction attempts from the thick, dense bone repeatedly failed. Then, in 2025, geneticist Xiaomei Fu, the same researcher who had first sequenced DNA from that original Denisovan finger bone back in 2010, managed to recover genetic material preserved in hardened dental plaque still clinging to the skull's teeth. The results, published jointly in the journals Science and Cell, finally closed the loop. Dragon Man wasn't a mysterious new branch of the family tree after all. He was a Denisovan, the first reasonably complete skull ever confidently identified as belonging to that population, giving researchers, for the first time in 15 years of study, an actual face to put to a lineage that had, until that point, existed almost entirely as scattered fragments and abstract genetic signatures. It turns out we had a Denisovan skull sitting in plain sight since the 1930s. Nobody realized what they were looking at until the DNA finally caught up and told them. That resolution matters directly for the puzzle at the heart of the Japanese archaic DNA story, the strange near-total absence of Denisovan fossils across most of Asia, despite their DNA being scattered through millions of living genomes. Part of the answer, really, is environmental. Much of East and Southeast Asia sits under warm, humid, biologically active conditions. dense jungle, acidic soil, monsoon cycles that flood and erode the landscape repeatedly. Exactly the kind of environment where bone breaks down within decades rather than surviving for tens of thousands of years the way it can in a cold, dry cave in Siberia or a frozen tundra in northern Europe. Most of the world's best preserved ancient fossils come from places where moisture and bacteria have as little to work with as possible. Large stretches of Asia are close to the opposite of that. But the Dragon Man case adds a second, more unsettling possibility to that explanation, that some of the fossils were never actually missing at all. They were simply sitting in collections, unrecognized for decades, waiting for the right combination of genetic technique and persistence to reveal what they actually were. If one dramatic, nearly complete skull could sit misidentified for close to a century, it raises a real question about how many other partial skulls and jawbones scattered across museum collections throughout Asia might also belong to Denisovans or to still unnamed relatives, without anyone yet having the tools or the reason to look closely enough to notice. There's also the matter of altitude, because one of the clearest, best documented examples of archaic DNA doing real, measurable work in a living population comes from Tibet. Researchers identified a gene called EPAS1, present in Tibetan populations, and closely tied to their remarkable ability to live and thrive at extremely high altitude, where oxygen levels most human bodies simply cannot adapt to comfortably. When scientists traced the origin of that specific version of EPAS1, they found it wasn't a new mutation that had arisen recently in modern humans at all. It was inherited directly from Denisovans, who had apparently already adapted to those extreme high-altitude environments across the Tibetan plateau long before modern humans ever arrived there. In other words, an extinct human population handed down through interbreeding a biological survival tool precisely suited to one of the harshest environments on Earth, and that tool is still actively protecting people living at high altitude in the region today. It's one of the clearest, most concrete examples anywhere in the archaic DNA field of exactly why evolution kept these ancient genetic contributions around instead of quietly discarding them. They worked, and in some cases they still do. So what does all of this actually mean? Not for researchers publishing in journals, but for the roughly 125 million people currently living in Japan, and honestly, for the rest of us too. For most of modern history, humanity told itself a fairly tidy story about its own uniqueness. One species, one clean lineage, the sole surviving branch of a much larger evolutionary tree. That tidy story simply isn't accurate anymore. Every population outside sub-Saharan Africa carries some measurable percentage of DNA inherited from at least one archaic human lineage. And in East Asia, specifically, that inheritance appears to be more layered and more genetically diverse than researchers assumed even a decade ago. Not one clean signal, but multiple overlapping ancient contributions folded into the same modern genome. This reality is no longer confined to academic papers either. Commercial DNA testing has quietly made it personal for millions of ordinary people. When someone orders a consumer ancestry kit and gets their results back, alongside an ethnicity breakdown, many now also see an estimated Neanderthal percentage, and a growing number of services are beginning to report Denisovan ancestry as well. People are learning, often for the first time, that a measurable slice of their own genetic code traces back to a population that vanished from the Earth tens of thousands of years before anyone ever wrote anything down. What that discovery means to people varies enormously. For some, it becomes a genuine point of pride, a distinctive, deep-time thread woven into their personal ancestry story. For others, it stirs something closer to quiet unease, the sense that a piece of who they biologically are isn't fully, cleanly modern, human in the way they'd always assumed. Neither reaction is wrong, because the underlying biology really is strange enough to sit with. We are, every one of us, outside deep sub-Saharan African lineages, walking genetic mosaics, carrying working active fragments of code inherited from separate human lineages that our species once lived alongside, competed with, and at some point chose, in whatever sense that word applies across tens of thousands of years, to have children with instead of simply staying apart from. That reality has started creeping into conversations well beyond genetics labs, into questions about identity, categorization, and what terms like ancestry and ethnicity are even supposed to mean once you accept that everyone's genome is a blend of contributions from multiple, once distinct human populations. Some researchers argue this evidence should push back hard against rigid biological categories altogether, since the genetic distance between any two living populations increasingly turns out to be, in part, a story about which extinct lineages happen to contribute to their ancestors, rather than anything resembling a clean, fixed. Others push back on that framing, pointing out that the specific effects of this archaic DNA on immune response, on metabolism, on disease risk, are measurable and real, not just abstract statistical noise. A Japanese individual carrying that Denisovan-linked NK61 variant may genuinely process a diabetes medication differently than someone without it. That's not a social construct. That's a biology doing something specific inside a specific body, and it has direct implications for how medicine gets tailored to different populations going forward. There's a more recent piece of research that adds an important layer of nuance to this whole picture, because it shows the story of Denisovan ancestry in Japan wasn't fixed from the very beginning. It kept shifting as later migrations arrived. In 2025, researchers analyzing ancient Jōmon genomes, including one individual who lived roughly 4,000 years ago, found something that initially seemed to run counter to what you'd expect. That individual carried only a small fraction of the Denisovan ancestry found in modern Japanese populations today. Comparing that against a much larger data set of over 100 ancient genomes from across East Asia, Siberia, and beyond, the researchers found that mainland populations living in what's now China and Mongolia carried the highest concentrations of Denisovan ancestry recorded anywhere in Eurasia. The implication is that later waves of migration, particularly during the Kofun period between roughly the 3rd and 8th centuries, introduced additional Denisovan ancestry into the Japanese archipelago from the mainland, gradually diluting and reshaping the genetic signature the earlier Jomon population had carried on its own. In other words, the Denisovan DNA sitting inside modern Japanese genomes today isn't a single, unchanged inheritance passed down cleanly from one ancient encounter. It's the accumulated result of multiple separate waves of ancestry layered on top of each other across thousands of years, each contributing its own piece to a genetic picture that's still being pieced together. The research itself isn't slowing down either. Scientists are increasingly moving past simply cataloging where archaic DNA sits inside modern genomes and starting to test what it actually does at a functional level, inserting archaic gene variants into laboratory-grown cells to observe how they behave differently from their modern human counterparts, comparing ancient and modern versions of the same gene side by side to map out the biological differences directly. Every year, sequencing gets faster and dramatically cheaper, and every year, researchers analyze more genomes from more populations, and the human family tree keeps getting more tangled rather than simpler. If Denisovans really were spread across Asia as multiple genetically distinct populations, as the Papuan research already suggests, and if a Denisovan skull could sit misidentified in a museum drawer for the better part of a century before anyone realized what it was, it's a reasonable bet that the current picture, tripartite Japanese ancestry, 44 archaic DNA regions, a newly faced Denisovan population, is still an incomplete sketch rather than a finished portrait. The Japanese genome study didn't close a chapter on human evolution. It opened one, and it's a chapter written directly into the cells of people alive today, carrying forward, without ever fully realizing it, the biological legacy of populations that vanished from the earth tens of thousands of years before recorded history even began.